From today's lecture you're talking about? So a couple weeks ago I have done an overview for that, so I'll refer you to that. Some people asked me if I would do a sort of a mini thing since the last overview, and I will do a little bit of that. Well, the overview is actually, no, the overview is right there, overview number 1. So the overview covers up through a week ago. So there's 3 new lectures since then. I'm going to talk a little bit about those here. Okay, I guess I will get started since everybody has got important things to do, like studying for biochemistry, right? You guys are planning to study, I hope. So I realized talking to some of the folks up front that I hadn't posted an old exam from 451, and 451 has a different format than 450. As I was telling them, this is a simpler format. It's a simpler format. It's not going to take you nearly as long to take an exam. It's got short answers. It's got some what will look like true matching, but they're actually true-false. Not matching, multiple choice, but look like true-false. Look like multiple choice, but are true-false. That's the way I should say it. And there's matching is the last thing. So it's a pretty straightforward format. But I want you to see the format, so I'll post that, and I'll put a note out to everybody announcing that as well. Yes?
Kind of a quick test, but are we going to have to explain why it's false?
Do you have to explain why something is false? Well, you'll see on the exam that I'm going to show you, no.
Sorry, Curtis in molecular bio, next field everyone on the test.
Well, if you're going to say something's false, you should know why it's false, right? I mean, I think I'm not going to do that, but I think that's not an unreasonable thing to do, because otherwise you're giving everybody a 50% chance of guessing it right. So if you say it's false and you have to say why, then that's kind of an assurance that you know what the question is and you weren't just guessing. That's why they do that. Yeah. Okay. So I also had a request from over here, and I'll ask the room to do a bit of an overview since the last overview before I take questions. Is that something you want? Is that you want to dive into the questions? What do you want to do? Overview since overview. There's a vote for that.
I would vote for overview
of today. You just want overview of today? That's what you guys said also, right? Or you said, right? You're saying no. You want overview since the last overview. Can't please everybody. Okay, let me do a quick overview since last Monday, which is when the last overview was through. Okay, so that means Wednesday's lecture, that means Friday's lecture, and that means today's lecture. Okay, so I'm going to do a quickie on that, and then I'm going to turn it to questions. So if I recall, well, actually I can look at the highlights. I can tell you where I started on Wednesday. That would have been electron transport. Okay, so basically at electron transport, okay, so basically starting with electron transport. What did I talk about? I've got, obviously, sources of biological energy largely through oxidation. Plants, of course, an exception to that with photosynthesis. And the photosynthetic fish that I talked about today also would be an exception to that. Oxidation, of course, is what's involved in redox reactions, and redox reactions are named that because for everything that is oxidized, something else gets reduced. So when molecules get oxidized, we have electron carriers that accept those electrons. And so you've seen, for example, this figure that I showed from glycolysis, where this guy, which is an aldehyde, is becoming, in this case, an ester, but it's oxidized to an acid, so that's an oxidation reaction there. And those electrons from here get transferred to NAD, which makes NADH. So, and we've sort of gone through that a few times now, that redox reactions always have a pairing, between the molecule oxidized and the molecule being reduced. Photosynthesis, of course, is the source, ultimate source of essentially all energy on the face of the Earth. The rare exceptions would be things that are using chemical sources or heat sources deep in the ocean, but for the most part, 99.999% of the energy on the Earth is coming ultimately from photosynthesis. Catabolism is paired with anabolism in the same way that reduction is paired with oxidation. And anabolism requires energy and requires ATP. And ATP is produced by catabolic processes. So anabolic processes are those that build molecules, as you see up here, starting with simple building blocks and going up to larger molecules. Catabolism, the breakdown of things and the oxidation of them, produces the energy necessary to make ATP, which makes that all possible. Okay. I talked about the Nernst equation, and the Nernst equation arose from the fact that we have an electrochemical gradient. The Nernst equation is an attempt to quantify that energy difference arising from unequal distribution of ions across a membrane. And the Nernst equation that I showed was right here. The Nernst equation came ultimately from reductive power, and the E in this term here is a measure of that reductive power. The reductive power is the tendency to take electrons away from things, and I talked about how reductive power is essential when we have things going down a chain being oxidized. So each one is taking electrons from the thing previous to it. Did you have a question? Okay. So as I said, you're not going to do calculations for this, but I think understanding that the Nernst equation does in fact allow a calculation of what that reductive potential is. And I showed how the delta G for a reaction takes into consideration that change in the reductive power between two different molecules. And so that F being a Faraday constant, and I didn't say what it was. I'll tell you here. It's just the number of electrons involved in the reaction. Okay. The measurement. So I showed these half-cell reactions, reference cell reactions, and I'm not going to have you draw that, but I just wanted you to see it. Okay, so getting to the nuts and bolts of the electron transport system, I talked about how that occurs in the matrix. In the inner membrane of the mitochondrion, and that's where the various complexes and smaller molecules are all located. And I pointed out the convenience of that because the citric acid cycle, of course, is occurring in the mitochondrial matrix, and all that NADH that's being produced and all the FADH2 that's being produced. What's that thing doing? Go away. I hate bumpy, jumpy things. Okay. Is then fed into the electron transport system. So the NADHs enter the electron transport system from the inside of the mitochondrion, and they are going through the various complexes in the membrane. I didn't say it, but I don't think I said it, but I will point out to you that NADH that's produced out in the cytoplasm does not make it easily into the mitochondrial matrix. Instead, it has to be shuttled in. There are molecules that actually shuttle the electrons in and then give them to NAD or FAD as appropriate. And then those NADHs and FADHs then pass things into the electron transport system. The electron transport system, as I talked about, is a set of complexes that are separated by small mobile type entities. I won't call them molecules, but entities. So if we look at the complexes, they're numbered as complex 1, complex 2, not shown on the one above, but there's complex 2 down here, complex 3, and complex 4. So those are all multi-protein complexes that aren't very mobile. They don't move around very well, but they are embedded in the mitochondrial inner membrane. You can see NADH being passing off its electrons to complex 1 here. And then complex 1 passing electrons through coenzyme Q to complex 3, to cytochrome C, to complex 4. And within complex 4, we see electrons that are donated to oxygen to make water. And oxygen, of course, is the terminal electron acceptor for the electrons that actually started up over here. The oxygen is necessary for the electron transport system to operate, meaning that if you run out of oxygen, as happens in some cases, then the electron transport system will stop, and that's why it's important to have fermentation as a backup whenever you have that situation happening. Complex 2 is the equivalent, or not the equivalent. Complex 2 is the succinate dehydrogenase that we talked about in the citric acid cycle, and you can see it's embedded in the membrane. it actually can accept electrons from FADH2. Okay? And so that's why we call it complex 2 and why it's actually a part of the system right here. Let's see. Let's keep going. I'm on a roll here. 1 and 2 don't interact at all. No. And keep in mind that there are thousands of these on any single mitochondrion. So people said, well, is this order, does it always go in this order? And I say, well, think about it. I mean, coenzyme Q, for example, isn't tied to any of these. We show it going to 1, but complex 2 is bouncing all around. If it bounces, it has no electrons, and it bounces into complex 1, complex 1 is going to give it some electrons to go off to complex 3. But the complex 3 it takes it to wouldn't necessarily be the same one as before. We just show them as sort of linear arrangements in here. But the reality is these are all over the intermitochondrial membrane. Yes? 1 and 2 both give electrons to complex 2, and both of them give electrons in pairs. Yep. Okay. I showed you this anatomy of the mitochondria. There's the outer membrane there. I showed the inner membrane is this sort of yellow thing, and you can see how it's invaginated all the way through here. And all those little infoldings of the intermitochondrial membrane increase the surface area. And increased surface area means that you have all the more places to put more and more complex 1s, 2s, 3s, and 4s. The matrix, of course, is the sort of blue area in the middle, and that's where the liquid, as it were, of the intermitochondrion is located. And that's also where all the enzymes of the citric acid cycle and fatty acid oxidation are all located. Okay. This is another schematic of the same thing, a little bit simpler. But this one now shows what happens as a result of action of the electron transport system, and that shows the pumping of protons out into the intermembrane space, as you can see here. And that means that the concentration of protons when electron transport is going on, the concentration of protons is higher in the intermembrane space than it is in the matrix, meaning the protons want to make it back into there. And that's important. Okay. Yeah? There are no proton pumps in the outer membrane, no. Okay, so the electron transport system goes. Here's the electrons from NADH going into complex 1, going to coenzyme Q, converting it from Q to Q, what I call QH2. That's the reduced form, and that's the oxidized form. The Q pool, or that is these coenzyme Qs donate their electrons to complex 3. Complex 3 donates them to cytochrome C, and the cytochrome C donates them to complex 4, and 4, of course, donates them to oxygen to make water. Now, in the process of moving through complexes 1, 3, and 4, you see protons being pumped outwards, which is how the proton concentration gets higher on the outside than on the inside. And complex 2, which is coming up, complex 2, okay, basically follows the same path, but you see now they're passing electrons off to coenzyme Q, but no pumping of protons. So there's no protons going through complex 2. So I had a question from a student on the eCampus today saying, well, how come they say that there's no energy from complex 2? No, that wasn't the question. It was how come there's no protons pumped here, but you get energy from FADH2? And the answer, of course, is because the electrons ultimately make it to these two and still generate a proton gradient. It doesn't generate as many protons, but it does generate some.
Complex 3 in this one looks like an integral protein.
Yes, it is.
The other figure is more like a peripheral one. So
Which one?
one before was the
That's complex 1. The green one. Oh.
It's kind of like more peripheral.
But it's definitely an integral membrane. Okay. Protein. Yeah. So just bad art. Okay. So those show the overall complex there. Coenzyme Q has the ability to exist in 3 oxidation states. And that means it can have 2 electrons, it can have 1 electron, it can have 0 electrons. And that turns out to be important because coenzyme Q is what I referred to as the traffic cop. It's able to accept electrons in pairs, but it passes them off one at a time. And of course, that's what happens in the Q cycle that I talked about. So the Q cycle occurs in complex 3. And the Q cycle looks like this, okay? Now, as you'll see when I put the things on the highlights, and you see up here, putting these things into words makes it a bit more complicated than the process actually is. The process occurs in complex 3, and complex 3 is what I described as a docking area for these various things to come in and dock, the coenzyme Q and the cytochrome C. So the cytochrome C is docking on the top. Coenzyme Q is floating around in the inner mitochondrial membrane. And the process starts with an oxidized coenzyme Q, a reduced coenzyme Q and an oxidized coenzyme Q. And people say, where do they come from? Well, there are billions of them floating around the membrane. Some of them are oxidized and some of them aren't, right? And so one that's oxidized pops in down here. One that's not oxidized goes up here. And that's all that's happening. Once it's there, the process starts. The 2 electrons from the top one split. 1 electron goes down here. 1 electron goes to cytochrome C. The protons get split off, and the protons ultimately get pumped out. You can see that happening right there. Okay? When that happens, then the Q that was there, which is shown here, pops out. It's fulfilled its function. It doesn't have any electrons. It pops out and goes back out here into the membrane, where ultimately it can be picked up by something else, right? This guy, which got an electron, is holding on to it. It's what I show here as Q minus. It's holding on to that 1 electron. Okay? So that Q minus is holding on to 1 electron. In the second step of the Q cycle, a second QH2 comes in. That's a reduced coenzyme Q. And the same thing happens. 1 electron goes to a cytochrome C. This first cytochrome C, when it got its electron, took off, right? Another one came in and grabs the second electron. Now when you put the second electron down here on this guy now has 2 electrons. Adding an electron to a Q minus gives you a Q minus minus. Okay? So it's the fully reduced form, and the Q minus minus really loves protons, so what does it do? Grabs 2 protons and becomes QH2. So that's what's happening in the Q cycle. Very simple. Just splitting is all that's happening each time. Yes, ma'am?
Are the docking stations ever considered empty, or are they just like constantly?
Sure. They'll be empty sometime. Yeah.
Like both of them? Or
Sure.
Okay.
Sure. Yeah. Yeah. Yes? Yeah. Yeah, so once it's gotten oxidized, it can go back to complex 1, sure. Or it could come in to an open site of this and perform the same function, right? So Q is there to do anything that, whatever it happens to bump into next. So as I said, there's millions of these things in a membrane. And so where it happens to bump into, that's just random in terms of what the next thing is it goes and does. Once the bottom Q what? Once the bottom one becomes QH2, yes, it exits, right? So then it's got to what? It's going to come back up and find a top one somewhere to go into. Okay? Well, in the process of leaving, it's getting the protons. The when and where doesn't really matter. The point is it gets the protons. Yeah.
Is there something there that's keeping it there until it's Q minus though?
Q1 minus never gets released, no. So it's not going to come out of there until it's Q minus 2 and then getting the protons on the way out. So Q minus 1 is not going to come out. Okay. The Q cycle. Cytochrome C, as I said, was a protein that is conserved from bacteria to humans. It says a lot about its importance in terms of carrying electrons. You see it here shown as a peripheral membrane protein. This is a true peripheral membrane protein. I talked about how when damage occurs to the mitochondrion, one of the things that can happen is cytochrome C can be released out into the cell, and that is a trigger for the cell to recognize it's time to commit suicide. It goes through the process called apoptosis. Cytochrome C is fairly small protein, and it's very mobile. So just like this guy is small and mobile and goes back and forth very readily, this guy's mobile and serves a nice shuttle function. Okay? Yes? There's a heme group in it, yes. That right there, yes, that's a heme group, yeah. So whenever you hear the word cytochrome, it's got a heme. Yep. There's a metabolic melody, actually. I've never finished. I started this years ago, and maybe somebody can help me with it, okay? So does anybody know the old Paul Simon song called Kodachrome? No, I'm an old guy. All right. I'm asking the wrong group. All right, never mind. Never mind. Okay. Was that a question?
Yeah. So controlled electrons can only use. Oh no,
No. Cytochrome C can only handle one electron at a time. Because what happens here, this takes 4 electrons for this guy to get over here. That means 4 different cytochrome Cs have to come in to complete that process. And it's this process that gets interrupted sometimes. So if it gets interrupted and doesn't complete its cycle, then what's going to happen? You're going to have a reactive oxygen species. That's one of the ways of generating reactive oxygen species. Okay, complex 4. Complex 4 is where the magic of the electrons reaching their destination occurs. I pointed out that cytochrome C comes in reduced, goes out oxidized. There are 4 of these to make 4 electrons, and it takes 4 full electrons to make that happen. The electrons pass through cytochromes that have hemes within cytochrome that contain copper in one case, or actually in 2 cases, and iron in 2 cases. And so there's just a movement of electrons through there. And to have something that is able to carry an electron, if it's a metal, it has to be able to exist in 2 different oxidized states. So for copper, that's +1 and +2, +1 being when it has the electron, +2 when it doesn't, reduced to oxidized. And iron +2 to +3, +2 being the reduced form with the electron, +3 being the oxidized form without it. Well, yeah. Well, that's basic chemistry, right? So, I mean, anything basic chemistry is fair game, I think. So, all right. So, pumping of protons also happens in complex 4. As I said, I'm not expecting you to memorize how many protons. I think people get too weirded out and obsessed with numbers of protons, including the people who do the research on this, to be honest with you. But the point is protons are getting pumped, helping to make that proton gradient, which makes the ATP. The electrons get dumped onto oxygen to make water. 4 protons, 1 molecule of molecular oxygen, and there you are. Yes? Uh-huh. So if you didn't completely reduce this, you're going to have at some point, you're going to have an oxygen that doesn't have the 4 electrons that it needs. So I didn't show you the whole process, but since you've asked the question, I'll bring it up. The oxygen comes in as molecular oxygen, which means the 2 oxygens are bound to each other, right? It's a covalent bond between the 2 oxygens. You add 1 electron to that, you've created an oxygen minus, okay? That is a superoxide, right? You could put a proton on there, but now you've got a molecule that wouldn't be stable, HO2, for example, that wouldn't be stable out apart from here. So the second electron comes in and now makes an HO2 minus, right? And that will also grab a proton. Well, if you do that, you've made hydrogen peroxide. Hydrogen peroxide, as I talked about, is also a reactive oxygen species. So incomplete reduction of this will lead to reactive oxygen species. That's the point. So ultimately, you will the last 2 protons break the bond between the 2 oxygens, and then you get an H2O and an H2O. Okay? But I'm not asking you to understand the steps in that process. Okay, I meant to talk about it. I didn't talk about this guy, so I'll show it to you here, but you're not responsible for it. But this is kind of a cool shortcut that occurs in plants, fungi, and protozoa. And so what happens with this is they have a protein that is actually able to reduce the oxygen to water. And you might say, well, why would you want to do this? Because you're not going to get as many ATPs. And when you see a short circuit like that, it means that the cell has to burn more to make the same number of ATPs. And what happens when you burn more? You increase heat. And so this is actually used in some organisms, not by us, but in some organisms as a way of generating heat. I didn't show you in class, so you're not responsible for it. Okay, so the requirements for electron transport, you have to have electron carriers that are reduced, these 2 guys here, and you have to have oxygen. If you're missing either one of those, you got trouble. All right, I talked about inhibitors. Rotenone and amytal inhibit complex 1. I pointed out they weren't quite as poisonous as some of the ones that inhibited the other ones. Antomycin inhibits complex 3. And then I pointed out other inhibitors and where they inhibit. So here's the rotenone and amytal inhibiting right here, antomycin A inhibiting right here, and cyanide, carbon monoxide, and azide inhibiting down here. Okay, so this is why cyanide and carbon monoxide are very, very poisonous to you. And there's no real way to bypass this. So if this plugs, anytime you put a plug in here, I put a plug right here, everybody stops. And when everybody stops, what happens to the concentration of NADH? It goes up. NADH goes up, citric acid cycle stops, you got problems. So, and that's true whether I stop here or I stop here. Okay, let's see. What else did I say? Briefly. Reactive oxygen species. Okay, so reactive oxygen species happen or have a variety of structures. One easy way is to have a free radical in which we have an unpaired electron, and that causes major problems because such a molecule will instantly react with the first thing it bumps into. And I pointed out that if you bumped into a guanine, for example, in DNA, you can make 8-oxoguanine, and 8-oxoguanine will base pair with adenine. Makes some real big problems for mutation. Superoxide, which is this compound I showed here, results from addition of one electron to a molecular oxygen. You combine that with nitric oxide. Nitric oxide is used by our cells for signaling purposes. You create a molecule called peroxynitrate, which has some really nasty properties, very, very toxic compound. And this guy right here is also extraordinarily reactive, and it will do things like nitrate tyrosine that I talked about. And I pointed out how nitrating tyrosine could have some very undesired effects by changing the charge on tyrosine and maybe affecting signaling and a variety of things. There's some interesting evidence that this may play a role in the nitration of tyrosines in nerve cells and perhaps play a role in Lou Gehrig's disease. Yes, sir?
Do we have enzymes that can neutralize peroxynitrate, or is it kind of game over?
You know, to be honest, that's a very good question. I don't know of enzymes that will do that. The enzymes that we have are focused here. So that's a very good question. I'll have to look that up. I don't know the answer to it. So the protection that we have against reactive oxygen species, of course, you always have antioxidants for things like, let's say, peroxynitrite or something, so that would provide some help. But the reactive oxygen species antioxidants, these are chemicals that will bump into and basically disable reactive oxygen species, handling that electron and basically converting it into a form that's much less reactive and much less problematic for cells. The other way to do that is with enzymes. And so I talked about catalase and superoxide dismutase. Catalase is involved in converting hydrogen peroxide, H2O2, into water. And that's the reaction that it catalyzes. It uses glutathione as a way of donating electrons to reduce this guy further and releasing molecular oxygen. So we go from a reduced glutathione to an oxidized glutathione. Yes, ma'am?
What did guanine turn into when it got...
What to what?
Guanine turn into?
Guanine will turn into 8-oxoguanine. Okay, puts an oxygen on position number 8, and that will make something that can base pair with adenine. Not good. I was surprised nobody asked me in class what causes this to go back. And I'll tell you here, you don't need to know it, but it's reduction by NADPH actually will cause the oxidized glutathione to be reduced back to reduced glutathione. Yes? Glutathione is a tripeptide. It's a very unusual tripeptide. It's right here. Glutathione looks like this. That's actually 3 amino acids that are joined together. That's actually a cysteine right there. Okay? And this is one of the very, very, very few peptides in cells that's not made by a ribosome. It's actually chemically synthesized within cells. Okay? But the importance of glutathione is actually that sulfur, which comes from the cysteine, because that's the source of electrons. This is the reduced form of glutathione here. When you oxidize glutathione, you join 2 sulfurs together. The electrons pop out and go to the acceptor molecule, and this leaves you with the glutathione in the oxidized form. So it takes, obviously, 2 of these to make 1 of those. I know I'm going fast, but if I don't, we'll be here all night. Okay, superoxide dismutase is an enzyme that does a ping-pong reaction. Somebody online asked me, well, what if it just did the copper reaction, it wouldn't have to ping-pong and it would be faster? The answer is no. You can do that one reaction, like, let's say, converting this guy to this, okay? But then you've got superoxide that's been changed. It's still holding an electron. Remember, it's grabbing an electron from this guy, all right? And when it grabs an electron, the enzyme is no longer in its original state. You've got to get it back to its original state, and that's why the ping-pong is necessary. So this is an overview. I'll show you the reaction in just a second, but the overview shows the superoxide dismutase grabs the electron, okay, and it makes, in the first step, an oxygen molecule, gaining an electron, and then that oxygen, a second superoxide comes in. The electron on the superoxide dismutase is donated to that molecular oxygen, and hydrogen peroxide is produced. Hydrogen peroxide is converted into water and oxygen by catalase enzyme. Now, if I show you the ping-pong mechanism itself, easier to look at than it is for me to describe it, here are the two steps to the ping-pong reaction of superoxide dismutase. First step, that electron from superoxide is grabbed by the enzyme. Notice the copper plus 2 is becoming copper plus 1. And oxygen is released. This is where the student said, well, why do you have to do the second step? Because you're releasing oxygen, right? But the problem is you got copper in the plus 1 state. You got to get it back to the plus 2 state, or you would use the enzyme exactly once, and it would never function again. So the second step of the process, a second superoxide comes into the enzyme. You see the enzyme which had an extra electron donates the electron to the molecular oxygen. You can think of this as being O2 minus minus, and then that's going to combine with two protons to leave you H2O2. And that's how we got the hydrogen peroxide in that last figure that I showed you. And the enzyme now is back to its original state, copper plus 2. Okay. Let's see. What else do we have here? Okay, oxidative phosphorylation. Peter Mitchell, the guy shown here, came up with the chemiosmotic hypothesis. It required several things: an intact intermitochondrial membrane. It required movement of electrons that pumps protons, which we've already talked about. And it says the proton gradient drives the formation of ATP. It was a radical idea. It said that formation of ATP didn't come from a high-energy intermediate, but rather came from this complex that's letting protons flow through it. As I said, he turned out to be completely right and won the Nobel Prize for it. Back to our original figure, you see the movement of the electrons through the various complexes. You see the oxygen accepting the electrons and becoming water. And you see the proton gradient forming. So the proton gradient is necessary for this complex in yellow, which is the complex 5, or the ATP synthase. And the movement of protons through this complex is what drives the synthesis of ATP. How many people have looked at the video online that I linked to it? A couple, okay. It's a beautiful process, absolutely beautiful process. The amazing complexity. This is complex 5 in the intermitochondrial membrane. There's the intermembrane space above. There's the matrix down below. In the beta subunits, there are 3 of them, one here, one right back there, and one right there. That's the places where ATP is made. And there's also a little thing in the middle called gamma. And gamma is a little rotor. Just rotates like that. And you can think of it as pointing to any one of these 3 things. As it rotates and as it points, the configuration of those 3 beta subunits changes. And they have 3 configurations that can exist in what we call the L, T, and O states. I've yet to find a perfect figure for this. So I prefer to explain it instead of trying to say, well, look at the figure if you don't understand it. Any part, so these 3 beta units are here, 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 in color, okay? Each beta unit will have a shape that depends on the position. There's the gamma rotor in the middle. You see how it's turned? See the little pointer right there? See how the pointer is changed? See how the pointer is changed again? The movement of that pointer changes the configuration of each of those. So if we look, for example, at this guy here, it starts out as the O, meaning it's open and it's let out the ATP. The L which is converted to here, has accepted ADP and inorganic phosphate. And in the next step of the process, actually shown over here, the T, these guys are being squeezed together to make ATP. So this guy has gone from O, L, T. Okay? I usually say LTO, but LT, the next one would be O, because it's going to open to let that guy out. But it doesn't matter. O, L, T, or TLO, all right, same thing, is the progression of any individual one of these. Look at this guy. It's going from L to T to O, right? Or you look at this guy. It's going from T to O to L, right? So it's going from opening up, right, to binding, to squeezing together, to opening up, to binding, to squeezing together. And that's what's happening. So this process is very efficient. This process makes ATP. And as some of you have pointed out to me, the ATP synthesis occurs in the matrix. But the ATP is needed out in the cytoplasm. How does it get out there? Well, I didn't say that in class, but I'll tell you. It's not a big deal. There's a shuttle. And what the shuttle does is it swaps an ATP for an ADP. So the ATP goes out into the cytoplasm, and the ADP comes in. It's an antiport. Doesn't take any energy, and the molecules move across. Yeah. Yeah. This process is oxidative phosphorylation. That's right. So everything that's happening in complex 5 is oxidative phosphorylation. Okay? And that's actually a good question, because I actually distinguish these. Electron transport is what you see happening in complexes 1, 2, 3, and 4. Oxidative phosphorylation is what's happening in complex 5. Okay? Yep. Yep. Okay. And blah, blah. I talked about this having an F0 portion that's embedded in the membrane, an F1 head. I said the F1 head looks like a mushroom that's sticking into the mitochondrial matrix. As I said, anything I talk about in class is fair game. Yeah. Okay, there's our little video. Hopefully that helps. I find I get more confused by looking at those than I do anything else, but there you go. Okay, it'll go on forever. All right, I'm not going to show the video. I will talk about the requirements for oxidative phosphorylation. They include a proton gradient and ADP. If you don't have ADP, everything stops. If you don't have a proton gradient, everything stops. All right? Very important things for oxidative phosphorylation. Peter Mitchell said you had to have an intact mitochondrial membrane, and you do. If you don't have an intact mitochondrial membrane, as I said today, the likelihood you're going to make very much ATP is very, very low. Okay, oligomycin, inhibitor of ATP synthase. If I have that in my cells, I'm going to stop the oxidative phosphorylation. What happens if I stop oxidative phosphorylation? Let's go through a practice one. Stop oxidative phosphorylation. What happens? So I'm going to stop electron transport. How? Why does electron transport stop? Proton gradient gets too high, right? Proton gradient gets too high. There's too much water I'm trying to push uphill. I can't push any more water uphill, right? Proton gradient is too high. Electron transport stops. What happens to oxygen use? Goes down, right? What happens to NAD concentration? I hear dissent. Up or down? NAD is going to go down because you need to have electron transport going to make NAD, right? So NAD is going to go down. NAD goes down. What happens to citric acid cycle? Goes down. What happens to glycolysis? Goes down. Kind of like the stock market. Goes down. We just put an inhibitor in there, and we know what happens to the stock market. Okay, respiratory control. I said that, what did I say here? This was sort of what I summarized in the things that I talked about there. I'm not sure I actually showed this figure in class. And I show you these again, not to give you something to memorize, but you know, if you want to kind of test yourself and think about things, these can help you with that. Yes?
So last night, where it had a requirement for oxygen,
Yep.
oxygen was a
Oxygen is needed for electron transport. Right? So electron transport is what makes the proton gradient, which is the ultimate product of electron transport. Right? So that's why I didn't put that on there. There's a lot of things. I could say You can't have an inhibitor present, right? Because if I have an inhibitor present, I'm not going to have oxidative phosphorylation either. But the two things that you have to have is a gradient by whatever mechanism and ADP. Okay? Good question. Okay, let's see here. Tight coupling. I talked about coupling. So what's coupling? Coupling means that you have an intact mitochondrial membrane, and because of that, oxidative phosphorylation and electron transport are intimately linked. Okay? They're separate, but they're linked. One depends on the other. You can't have oxidative phosphorylation without electron transport, and you can't have electron transport without oxidative phosphorylation for the reason I just stated. The proton gradient gets too high, right? If you uncouple, you poke holes in the membrane, you destroy the integrity of the mitochondrial inner membrane, what happens to the gradient? Gradient, bye-bye. When gradient bye-bye, what happens to oxidative phosphorylation? Okay? What happens to electron transport? Going through the roof, because nothing stops it. No matter how many protons you pump out, they come right back in, right? So no gradient, no control of electron transport. Electron transport goes like crazy. Citric acid cycle goes like crazy. Fatty acid oxidation goes like crazy. You do lose weight in your sleep. A deadly amount. Okay? There are many things called uncouplers. The one I talked about is called 2,4-DNP, 2,4-dinitrophenol. And the 2,4-dinitrophenol pokes holes in the mitochondrial inner membrane. That was the miracle diet drug of the early 1900s. Okay. That brought me to the rapid movement through today's lecture, which was found right here. And specifically, it talks about how we can make phosphatidic acid. Phosphatidic acid, I said, was important because it is a branch between the synthesis of fat and glycerophospholipids. Yes, ma'am?
First, when something is uncoupled, that would only be like if you're trying to kill the cell or something. Like, there's no reason that a healthy cell would.
There are actually reasons that a healthy cell will uncouple. Would you like me to give you a thought question for the exam? Okay. So, tell me a reason that a healthy cell might uncouple. Based on what I've told you already. Yes? Create heat. So it turns out that when you hear about things like brown fat, anybody ever heard of brown fat? Okay. Brown fat is really desired by a lot of people because it's a good way of burning fat. Brown fat, in adults, we don't have very much. In infants, there's a fair amount. But when we look in adults, the place where we see brown fat, it's located in the areas, you're not responsible for this, by the way, all right? It's located in the areas around our spinal cord. Generates heat. If there's something you want to keep at a relatively constant temperature, it's your nerve signals going through. You don't want to be like the snake. When you cool it down, it... Okay. You don't want to have that, so you want to keep it warm. So brown fat generates heat. How does it generate heat? It generates heat by using this really cool thing called uncoupling protein. And what is uncoupling protein? It drills a hole in the mitochondrial inner membrane. So what happens? Cells start generating a fair amount of heat, burning all kinds of stuff up, right? And then there's a thing that'll plug it up later. So cells actually have, in brown fat, they actually have that ability to do that. So I wouldn't say never, no, okay? But you don't want to mess with it, let's put it that way. Yes? Why are reduced electron carriers what? Not what?
So, like, if it's uncoupled, then the electron transfer is still healthy.
Yep.
Why is that not affected by?
It is. I'm not sure I understand your question.
So the idea that the NADH you get, like,
Oh, you're saying, do you run out of NADH? You say, oh, okay. Well, you would, but when your NADH levels falls, what happens to the citric acid cycle? It goes faster. And what does it produce? NADH, right? So that's, again, thinking back to exercise. Why is exercise important? Because exercise is favoring you burning things up. Citric acid cycle and fatty acid oxidation, right? And why are you burning things up? That'd be a good question on the exam. Well, because your ADP levels are high. When you exercise, your ADP levels are high. And high ADP levels means oxidative phosphorylation. And oxidative phosphorylation means electron transport and reduced proton gradient, right? Make sense? Okay, good. All right. But I diverge. Yeah, okay. So, synthesis of phosphatidic acid. This guy is a branch point, the synthesis of fats and glycerol phospholipids. Very simple synthesis. We start with glycerol 3-phosphate. We put 2 fatty acids on it. We got phosphatidic acid. If we take phosphatidic acid, we can go to make fat with it. Very simple reaction. Or we can go down here to make glycerol phospholipids. Another simple reaction. I didn't say the enzymes in class, but if you'd like, I could make you know them. I haven't written the exam yet, so I could add that. It's still possible. Would you like me to put that? If I said yes, then you guys would say, well, we know it's going to be on the exam, right? Okay. This is the regulation of fat breakdown. Fat breakdown is controlled by this signaling pathway that you've seen before, and it goes through control of a single enzyme, this single enzyme being triacylglycerol lipase. That enzyme gets activated by this signaling system that you see, and that's consistent with the other things that we saw. Epinephrine was favoring release of glucose, right? So that cells had plenty of energy to do stuff, breaking down glycogen, for example. Here we're breaking down fats to release fatty acids to make more energy, just like for the same reason we're making glucose. Okay. Where am I at? Fat synthesis, okay, which is what I got distracted from. Fat synthesis is simple. You take the phosphate off of phosphatidic acid. It leaves you with diacylglycerol, and you put a fatty acid on position 3, and you have a fat or an oil. Okay. General structure, you've seen that probably about 10 times now. I won't go through that. And phosphatidic acid add-ons, you've seen those a few times, but you see the X is where they can be added on. This whole brown and green and what carbon backbone that's in the middle there, my battery is running out of this, not much left to it. Everything on the left is the phosphatidyl part. The X is the add-on part. And so you have, if it's an H, it's a phosphatidic acid. If you put a serine on it, you get phosphatidylserine, etc. And I pointed out that these guys can be synthesized by 3 different strategies. One strategy is to activate the phosphatidyl part. So you put a CDP onto where that X is there, and you've got a CDP diacylglycerol. So you've activated the phosphatidyl portion of the molecule. If you have that structure, then you can take that CDP diacylglycerol, you can add an inositol to it, and you get phosphatidyl inositol, and you split out a CMP. This guy's an activated intermediate. Activated intermediates donate energy, okay, to add a part of themselves onto something else. So the energy is right here. You break that bond. The part that you donate is this part, and you see that they end up being joined through that phosphate. Yes?
Why isn't that activated intermediate not known as phosphatidyl CDP?
It could be. It could be. Yeah. If you called it phosphatidyl CDP on an exam, I'd say fine. Okay. Yep. Does that make you feel better? Does that make you feel better? But yeah, I mean, you're right. Based on what I've told you, there's no reason it couldn't be. I mean, it could be named that. Cardiolipin. Oh yeah. Question, Megan. I'm sorry. What's breaking the bond? This is an enzymatic reaction. So the enzyme is catalyzing that addition. So you're asking about this breaking right here? Yeah. So this is just an enzyme that's catalyzing that reaction. So it breaks it and then transfers it over to here. Okay? Okay. Okay. Activation of the add-on. That's the other way. So instead of activating the diacylglycerol part of it, you can activate the add-on. Here's the add-on. Phosphorylethynolamine. You've made an activated intermediate of the ethanolamine. Okay? So now the enzyme can add that onto a diacylglycerol. And when it does that, there's the CDP ethanolamine plus diacylglycerol. We get phosphatidyl ethanolamine. And there's our friend CMP that's split out. Okay, so that's the second way in which a glycerol phospholipid can be synthesized. The third involves swaps, and that's shown in the figure on the right, where we have a phosphatidylethanolamine that's swapping the serine for the ethanolamine to make phosphatidylserine and ethanolamine. Okay? This guy is another, and they're actually, I didn't say much in class about this, and I said, well, this is kind of an exception. It's not totally an exception. There are a variety of compounds that can be made by chemically altering the add-on. So this is chemically altering the add-on. I didn't want to go into that, but that's what's happening. You see a decarboxylation reaction. You can make phosphatidylcholine, for example, by putting a bunch of methyl groups on the end of this. Okay, but again, you don't need to know that. So. Okay, and let's see. Sphingolipid biosynthesis comes from serine and palmitoyl-CoA. It makes this 3-keto-sphingonine. Okay. And I don't care if you know the name of that. I don't care if you know the name of this. But you know that we make intermediates that are necessary. This guy right here at this point has one fatty acid tail hanging off of it, that tail that came from palmitoyl-CoA. To make a ceramide, I have to put another fatty acid tail on there. And that's actually shown right here. Oh, sorry, I jumped ahead to one. That's actually shown right here. So we see the addition of a second fatty acid tail. That's that R right there. My pointer is dying. Very fancy. I like this stick. Okay. There is the other fatty acid tail. So we see two tails sticking off, and that's shown schematically right here. And again, that's like the glycerol phospholipids, which also has two tails. It also has a polar end, but in this case, nothing has been put onto there. That's simply a hydrogen at this point. And so it's not really fully polar at one end. That happens with the addition of a phosphate compound or of a sugar. And that's what happens in the synthesis of these molecules. So if you add either of these phosphorylated compounds to that ceramide, you get a sphingomyelin. If you take that ceramide and you put a single sugar in the place of the R group up here, you get a cerebroside. And I love this one. If you put a complex sugar onto the R group, you make a ganglioside. The gangliosides are pretty hairy-looking structures. I pointed out that sphingolipid breakdown, if it's inhibited, causes some severe neurological problems. And that's just a summary of some of them right there. So you don't want to have any of those enzymes deficient. Cholesterol, yeah. Palmitoyl-CoA and serine. It's palmitoyl-CoA. Because that's the palmitoyl part is the part that gives that long tail. And acetyl would only give 2 carbons. Cholesterol comes from acetyl-CoA. There's the acetyl part of acetyl-CoA. We can track every frigging carbon in that molecule, where it came from. Every single one we know comes from acetyl-CoA. Pretty amazing. Starting with acetyl-CoA. 2 of them. There's the thiolase I talked about at the very end of the class, right? In the song. Thiolase. It's the same thiolase that breaks down fatty acids in fatty acid oxidation. We'll talk about that later. Okay? But that's what thiolase, where thiolase is important. You saw this earlier with the synthesis of the ketone bodies. This is exactly the same as ketone bodies. Thiolase catalyzes the formation of this 4-carbon compound. Add 2 more carbons, and you get a 6-carbon compound, HMG-CoA. HMG-CoA is converted into mevalonate on the pathway that leads to cholesterol. HMG-CoA is converted into ketone bodies in this pathway down here. Since we're following the pathway moving upwards, this enzyme becomes relevant because HMG-CoA reductase catalyzes the reaction that now favors the synthesis of cholesterol. HMG-CoA reductase is the enzyme that's feedback inhibited by cholesterol, and it's the target of drugs known as statins, which inhibit it. And the drugs that inhibit it look like HMG-CoA. They're competitive inhibitors of this enzyme. The point here is making these isoprenoid building blocks, or as they're also called, isoprenes. They have 5 carbons, IPP, DMAPP. And those two molecules come together to make higher order structures, as seen on the next figure, which is right here. 2 5-carbon compounds make a 10-carbon compound. Add another 5-carbon compound, you got a 15-carbon compound. Combine 2 15-carbon compounds, you got a 30-carbon compound. Rotate, twist, and make some bonds, and you got yourself lanosterol. Lanosterol is 19 steps away from cholesterol. Yes, ma'am?
So you mentioned the carbon compounds, did you mention all the names, but did you want to know all the intermediate names there?
Well, I think I did mention the names in class. I'm just kind of going through quickly here. Anything I talk about in class is always fair game. Yes? Mm-hmm. So I can't talk. I mean, this is a common question. Why has the system evolved this way? I don't have an answer to that question, but it has. And I will say that there are things that you can branch from here to go elsewhere. You can also branch from here and go elsewhere. Okay. So both of these are important intermediates in other pathways. That's part of an answer to your question, I guess. That when we go to make vitamin A, for example, okay, we break it off. I think it's the geranyl pyrophosphate. And vitamin E can also come off of this as well. I didn't show you those in class, though. But yeah, that's just the way it evolved. So I hope that, I know that's never a very satisfying answer, but that's it. Yes, Ron. Squalene, uh-huh. Yep. Well, it takes a second one to have that happen, yeah. Right. So it takes 2 of these guys to make that. Okay? Okay. So we get to lanosterol. As I said, it's 19 steps to get all the way up to cholesterol. A lot of energy necessary to do that. And so if you stop the pathway early, you save a lot of energy, and that's all good. I pointed out that cholesterol leads to bile acids. Bile acids necessary for emulsification of fat in our digestive system so that we can help digest it and also get it into the rest of our body. And I also point out that cholesterol can be made into the steroid hormones, progestogens, example of which is progesterone, mineralocorticoids, corticosteroids, androgens, and estrogens. And that's where I finished today. Yes, ma'am. Like this thing.
Like your body turns cholesterol into progesterone, which then converts into testosterone and estrogen.
Yep.
And they go down that pathway.
Yes. Yep. So if you're asking if I have progesterone in me, I do. Yep. Yep. Actually, there's another I ran out of time to tell you a really cool thing. I don't think I've told this class about this before. Have I talked about the importance of this in class? Okay. So I'll tell you here. You won't be responsible for it. But it's a cool story. This reaction going from here to here is almost absolutely unique in biology. We don't really have other places where we see catalysis that directly converts a non-benzene ring into a benzene ring. Okay? Well, why do we care about that? We care about that because the enzyme that does that is known as an aromatase. You're making an aromatic ring, and it's called an aromatase. Does that ring a bell for anybody about anything? I haven't talked about it in class. Maybe you've heard of it somewhere else. Okay? So aromatase catalyzes that reaction. All right? There are many tumors that are estrogen sensitive, meaning that the presence of estrogen stimulates them. You can actually tell pretty readily if you have a tumor that's estrogen sensitive. If you have an estrogen sensitive tumor, you don't want estrogen sitting around because the more estrogen you have, the more the tumor is going to grow because it's actually treating it like a growth hormone. Okay? Well, how do you then protect somebody or kill or stop a tumor that has estrogen, the way that you do it is you give them an aromatase inhibitor. Aromatase inhibitors stops this reaction, lowers the production, lowers the levels of estrogen in the person who has this enormously, okay, and stops the tumor from being stimulated to grow. So aromatase inhibitors are very important for that. Yes, ma'am. I think it's the orientation of the hydroxyl. Yep. Men have the ability to make this, but they don't make hardly any of this. They don't make hardly any of this, but I'm pretty sure that men do have aromatase. Yes. Yes.
So do the types of estrogen and testosterone differs?
Oh, of these two? Yeah, that's a good question. In terms of function, this is what really gives the male sex characteristics. This guy was big in the news in baseball about 15 years ago, when they had the steroid issue. One of the ways the steroids were given to these people was they didn't give them this, they gave them this. Well, this guy metabolizes into this, and so they were cheating, right? Well, you give somebody too much testosterone, you got some real problems, okay? One of which is your testes will shrivel up to nothing, which is kind of funny, I think. Right? But there's some other giant problems. But that one always kind of amuses me to think of, you know. So stay away from that stuff, guys. Anyway, I'm sorry, I'm getting off the subject there. But all right, now I've finished talking about the overview, and probably is a good point before I get into too much trouble here. I should take some questions. You guys, I know, had a bunch of questions, so if you ask too many questions, I just stick you with a stick. So.
What is the biological significance of like IP3 from PIP2? You said something about how the IP3 is water soluble and goes to the ER.
Okay.
Like that one.
What's the biological significance of IP3? And that's a signaling molecule. The only biological significance is what I said, which is it is soluble in the cytoplasm. So if I remember where I said that at, that must be up here, probably lipids and membranes. No, not that. Where's it at? Here it is. This is the one right here, right? Okay. This guy, okay, let's first of all just look at it. All right. It has a polar end. It has two nonpolar tails sticking out, so it's going to be stuck in the membrane. This guy is going to be facing the outside or inside. Actually, inside is relevant here. Inside of the cell. It's facing the aqueous layer. In the signaling process, okay, phospholipase C, actually, again, I'm telling you more than you need to know, but phospholipase C gets activated by a 7-TM, which is bound by a hormone. Hormone binds 7-TM, activates phospholipase C. Phospholipase C cuts this guy, and that's what creates the IP3, which is this guy right here, and leaves behind in the membrane this guy here. And my only point of showing this slide wasn't to reteach signaling to you, but rather to show you how this guy is nonpolar, is going to stay associated with the membrane, and this guy is going to go to the cytoplasm. And both of these guys turn out to be signaling molecules. This guy stimulates the release of calcium. This guy stimulates a protease, and they work together to stimulate that protease. But again, that's beyond what we talked about. Okay? So this guy is IP3, inositol triphosphate. Okay? This guy's diisoglycerol. Keep going.
Could you explain leukotrienes again?
Leukotrienes, okay? So leukotrienes are examples of eicosanoids. When I say eicosanoids, what should pop into your head? 20 carbons. That's what an eicosanoid is, 20 carbons, right? What do we make from eicosanoids? Well, first of all, what's the starting material for eicosanoids? Arachidonic acid, right? What do we make from arachidonic acid? That's kind of a circular argument, isn't it? We make eicosanoids from them. Okay, two classes. One class, prostanoids, right? Second class, leukotrienes. Both 20 carbons, both coming from arachidonic acid. What do we make from prostaglandins? Thromboxane, necessary for blood clotting. How do we prevent the synthesis of the prostanoids? And why does aspirin help to prevent that? It's inhibiting the COX enzyme, which is necessary to make the prostanoid. That's right. Very good. So I think I mentioned it briefly in class, but I'll say it again here. Where do you get arachidonic acid? You get it from glycerophospholipids in the membrane. There are lipases that cleave those guys off. You clip, the arachidonic acid is pretty chemically unstable, and it gets made into the prostanoids by the enzyme cyclooxygenase. Cyclooxygenase is inhibited by aspirin, also by ibuprofen, and other things called NSAIDs, non-steroidal anti-inflammatory drugs. Since the prostanoids and thromboxanes are involved in blood clotting, inflammation, and things like that, treatment with aspirin will reduce both of those. Okay? Make sense? Megan? I'm sorry, I can't hear it. Yes. No, actually, the ibuprofen has some effect as well. I don't think it's as strong as aspirins, but it will have some effect, yeah. Okay, yep.
Do you need to know the difference between the triglyceride and the triglycerol? And that one is like a fat, one's an oil kind of thing? Who's the
Ooh, who won?
No one.
So I use the terms interchangeably. Triglyceride, triglycerol, right? Glycerol, right? In fact, triglycerol really doesn't say much. So if you want to be more accurate, you would say triglyceride, right? Or triacylglycerol, that would be appropriate. But triglycerol by itself wouldn't really mean much. Okay? Well, I wouldn't use I wouldn't use triglycerol because I don't think it says anything. Triacylglycerol would be the same thing as a triglyceride. I tend to use the phrase triacylglycerol. Yeah. And I'll be clear, I'm not going to go pick what's the other name for triglycerol sort of thing. You know. There's a change in the highlight? Oh. Oh, no. Well, now if I'm going to stick somebody with this thing, I have to stand this much closer to them. I'll leave this up here for somebody else to enjoy. Okay. It wasn't intentional, no. So I don't have there's not a difference. I don't use fat versus oil for that. No. If I said that, that was not intentional. Yeah. Yes? Is there any adverse effects in the membrane? So is there, yeah, but the question is, are there any adverse effects for clipping this arachidonic acid off of the membranes? Not really. Not really, because A, there's not that much of it to have an effect, and you're still leaving behind one fatty acid on there, so it's still going to stay embedded in the membrane. It's not going to have a giant effect, no. Yeah?
Could you explain the relationship between cardiolipin and the inner mitochondrial membrane?
The relationship between cardiolipin and the inner mitochondrial membrane. Let's take a quick look at that. How about that? Cardiolipin, right there. Okay, all that this point is to show you is that cardiolipin is associated with cytochrome C. It's in the inner mitochondrial membrane, and when damage occurs, okay, you can see damage sort of schematically shown right here. These guys allow for the release of cytochrome C to go out into the cytoplasm of the cell and stimulate the process of apoptosis. That's the only thing. Cardiolipin is found almost exclusively in the inner mitochondrial membrane. And this appears to be its role in the inner mitochondrial membrane. Yes, ma'am?
Sorry, just to clarify, so cardiolipin is what releases cytochrome
see the damage right here? Going out. Yep.
So we don't need to know the specifics about how the process is so done.
No, but I'll show you here. How about that? Okay? So look at what's happened here. You see what that is? What chemical form is that? When you see an OOH. Well, no, it's not the OOH you see on a carboxyl. These two O's are joined to each other. In a carboxyl, you got a double bond O and then an OH. These two O's are joined to each other. What do you call that? Come on, chemists. No. Call it bad? Chemistry didn't teach you guys this? It's a peroxide. You didn't talk about hydrogen peroxide? HOOH? Let me draw something for you here, okay? HOOH. That is a peroxide. All right. How do you get a peroxide? Well, it is a reactive oxygen species, but how do you get it? How do we get from here to here? It is an oxidation that's happening. There's no oxygens here. Look at the oxygens. So, one of the real problems, one of the reasons that you have vitamin E for your membranes is to prevent oxidation that leads to peroxide formation. Okay. Now, if this had come up earlier, I would have made you guys absolutely responsible for it on an exam. I'm unhappy that chemistry has not taught you that. That should be a basic principle of chemistry. Come on. Not your fault. If everybody doesn't know it, it's not your fault. Somebody in chemistry didn't teach you something. Damn. I go kick somebody's butt. Whose butt should I kick? Who? You'd like me to go kick somebody's butt, right? Okay, other questions. I'm getting off the subject here. Other questions? What's that? Okay, I knew you had a question.
Whenever you are phosphorylating ADP to ATP, where does the free phosphate come from?
Whenever you're phosphorylating ADP to ATP, where does the phosphate come from? It's kind of like saying, where does the hydrogen come from when you've got a proton, right? So there's plenty of phosphate floating around in cells. And phosphate, you may remember I talked about vitamin D regulates calcium and phosphate levels. But free phosphate is quite abundant in cells. And for the reason, partly, is because it's constantly being broken down from ATP into ADP, right? So plenty of phosphate available. Yes? Are all the vitamins I talked about in class derivatives of cholesterol? No. The only one that is vitamin D. Vitamin A is not a derivative of cholesterol. Vitamin E is not a derivative of cholesterol. And vitamin K is not a derivative of cholesterol. But you can make A, E, and K from that geranyl-farnesyl pathway that I showed you. Cholesterol is the only one that comes, vitamin D is the only one that comes from cholesterol. So you learned 3 things that came from cholesterol, right? Bile acids, steroid hormones, vitamin D. Yes, Leo.
Cholesterol is like a form of fatty acid type thing.
Is what?
Cholesterol is like under the category of fat, kind of. Is that why ADP and K?
No, I would not put cholesterol under the category of fat. I would put cholesterol under the category of lipid. Fat is a triacylglycerol, right? So you would not call cholesterol a triacylglycerol.
Okay. But it's a lipid.
It's a lipid.
Is that why A, D, E, and K are considered fat-soluble vitamins? Because they're still of that pathway?
Right. So remember the definition I gave you of lipids to start with. The definition was the lipids will always have a portion of them that's very nonpolar. A, E, and K, and D for that matter, all have significant portions of them that are nonpolar, and that's why they are all fat-soluble. Right? Yeah. Yes, ma'am.
Would you like us to know the full names of E2 and E3 of pyruvate beta-hydroxy?
Oh, my God. I don't even know them.
Oh, okay, great. E2 and E3, good.
One of my rules is if I don't know it, you don't have to know it. Okay? You like that?
Yeah. One more.
Okay, that's fine.
Last one.
That's what I'm here for.
What about the double bond is important?
You get charged by the question. Did you know that?
No. What's important about
So succinyl-CoA dehydrogenase catalyzes the conversion of succinate into fumarate, right? That involves the removal of 2 hydrogens and 2 electrons. And the significance from what I said in class is that when we talk about fatty acid oxidation, you'll see an identical reaction that occurs in fatty acid oxidation.
Okay. Mine was about, maybe I misplaced the double bond, but I'm just...
The double bond is in the trans configuration.
Okay.
I said that, okay? But what happens with the reason that the cell is making the double bond is so that it can add an oxygen. And to add an oxygen, it adds water across the double bond very easily. That means hydrogen goes to one side, hydroxide goes to the other side. That's how you make malate. And this exact same thing happens in fatty acid oxidation. So we'll see 3 steps in fatty acid oxidation that are identical to those 3 steps in, those 3 molecules that are made in, actually those 3 steps that happen in citric acid cycle. So you see succinate to fumarate, fumarate to malate, malate to oxaloacetate. That involves double bond, addition of water, oxidation of hydroxyl. The same thing happens in the fatty acid oxidation. So you learn it here, you don't have to learn it there. Aren't you happy? Nobody's happy. What else can I tell you guys? This is interesting. I'm not getting that don't look at me look. Everybody's actually looking at me like I'm going to tell you something. Okay, so for the first question on the exam. Okay. It looks like this is a stopping point. All right. There we go. That always prompts a question. Yeah. Okay.
What is an overall question about the citric acid cycle?
Okay.
So no number of carbons, enzymes,
Names. Yep.
and then just a little bit of information about each.
Well, you know, if I said something about it, I mean, for example, alpha-ketoglutarate dehydrogenase, I said was very much like pyruvate dehydrogenase, and I said it used the same 5 coenzymes. I mean, that's an example. Wherever I said something that was relevant about it, yeah, sure. Well, if you look at what I said, the question was, you need to know where the intermediates are used, the anaplerotic versus cataplerotic pathways, basically, what you're saying. Yeah, well, again, if you look at what I said in class, this is what it always comes back to. I didn't go through those, right? So if I didn't go through them, I'm not going to go back and say, ah, ha, ha, this is going into serine, and you didn't know it, right? I mean, I don't get any joy out of that. So if I didn't talk about it, just because it appeared on the screen doesn't mean that I'm going to have you do it, right? I really, seriously only want to test you on the things that I talk about, which is why I always come back to, what did I say in class? That's the important thing, okay? Okay. All right, I want to see a high average on this exam, guys. Or I will be kicking your butts instead of the chemistry's professor's butts.